Okay, this is David Zierler, Director of the Caltech Heritage Project. It is Thursday, March 7th, 2024. It is my great pleasure to be here with Professor Barbara Imperiali. Barbara, it is wonderful to be with you. Thank you for joining me today.
You're welcome. I'm glad to be here.
Barbara, to start, would you please tell me your titles and institutional affiliations?
My current title, and I can explain it because it always sounds odd to people. I am the class of 1922, professor of chemistry and professor of biology at MIT in Cambridge, Massachusetts. The class, I always get the same corny joke where people say, "You don't look that old." And I say, well, it must be good, clean living or something. But anyway, it was a fund put together by the class of 1922.
And
I first moved.
are
Yeah,
you
go
the
ahead.
inaugural holder of that chair?
No, it's been around a while. The sort of head of the class that was responsible for the chair, I used to write him brief notes telling him what was going on, but I think he would have superseded 100 years old by now, and I think he was in a home at the time. So, uh, I actually moved to MIT on the Ellen Swallow Richards chair, so I'm happy to tell you about that later on.
Now, is it a dual appointment? Are you a professor of biology and a professor of chemistry?
Yes, that's right. Which is a nice thing for me. They are full appointments in both departments. When I first moved to MIT, I was professor of chemistry, and my work evolves and evolves, and I would have more interactions with the Department of Biology than chemistry sometimes. So biology offered me a second appointment where I would be secondary biology and primary chemistry. And then about a dozen years ago, I shifted to primary biology, secondary chemistry. So they're both real appointments. They're not sort of adjunct.
And this means that there are two department chairs to report to, two faculty meetings to go to. It's double everything for you.
Well, it felt like it was at the beginning until it seemed to make more sense to focus on the primary appointments. So right now, I report to the biology chair, Amy Keating, and I teach in biology, but I teach a joint class with my colleague Ron Rains in chemistry, so that's good. I stopped teaching organic chemistry when I moved to biology. That wouldn't have made any sense. And I have taught general biology and a principles of chemical biology upper level class since my move here. So my teaching actually does spread between the departments, but you can only go to so many meetings.
Barbara, in the way that you have shifted your primary appointment, I wonder how much of a stand-in that is for how your research agenda has changed. In other words, perhaps earlier in your career you might have called yourself a chemist who did biology. Are you now a biologist who does chemistry?
No, you know what? I'm still a chemist who does biology. It's funny though right now because to the chemists, I seem like I'm a biologist. I'm in a different building. Wow, it's scary. It's the bio building. They must have mice in the attics and in the basements. And to the biologists, I'm the go-to chemist. Like, I need to run this reaction. Can you help me? I've always followed my own mission of applying as much chemistry as I can into studying biology, but I really think like a chemist. I mean, the molecules are in my head, the interactions are in my head, but I've gained from being more resident in biology by having sort of a richer feel for the sorts of problems and things that are moving very rapidly and technologies in the study of different biological processes. So I feel I've gained from my transition over as primary in this department. I was trained as a chemist, so I come from that background. I always tell people you can't go uphill. You can go from quantitative and molecular down to a little less quantitative and less molecular, but you can't go uphill. You can't be more, sort of, I study little mice, so tomorrow I'm going to think about doing synthesis and organic chemistry. But you can roll down the hill a bit. even though you probably don't look very smart for a while. But that's okay. I don't really care about that.
Thinking about chemistry and biology, I'd love to gain your perspective on chemical biology as a discrete field, and particularly perhaps the influence of people like Dennis Doherty and you thinking about
Oh,
these
yeah, yeah,
things.
yeah, yeah. Oh, yes, of course. So it is so strange. This field is something that just at a certain stage wouldn't go away. So when I was a graduate student, you could either be an organic chemist or a biochemist. You couldn't be a chemical biologist, right? So, I patched together my training in a synthetic organic lab and then biochemistry lab of Bob Abels at Brandeis. So I evolved myself across those borders. Something in the middle there would have been chemical biology, but it didn't exist. And so then it started that the field, we began to name bioorganic chemistry the field, right? And what was that? It was sort of a lot more people developing model systems that mimicked biology. It wasn't what it is now. It continued and continued to evolve. There was a lot of molecular recognition work, a lot of model system works. But I think that there's a subset of organic chemists who are pretty fearless, including Dennis, who I deem as one of my best friends from Caltech, where we really weren't that afraid of trying to go over the border to bring our tools with us or our thinking with us to study, solve, bring in new hypotheses. Now, I did something which I only learned much later on in the day that my colleagues at Caltech thought I was completely crazy, which was quite soon after I moved to Caltech. I was 3 years at Carnegie Mellon. There was a little bit of an aborted faculty position, but then I started at Caltech new. And I was being invited to Gordon conferences, and one was called the Natural Products Gordon Conference. There were others called heterocycles, very much entrenched in organic chemistry. Only at every one of those meetings, they'd try to broaden the set of people by bringing in a few people who were more biological. So we were the biological, bioorganic chemists. And the one I went to, a group of us just got together and said, we're fed up of being parts of other people's meetings. So a group of us, Glenn Prestwich, or three of us, and Craig Townsend said, let us start a Gordon conference in bioorganic chemistry. Now, by the way, that term still holds, but it's morphed into chemical biology so that the field can define itself. Mm-hmm. And let's make the case to the Gordon Research Conferences that you need this because the field evolves and evolves, and the people who are involved in it need to talk to each other and need to evolve together and understand. And we need places for young people to go to meet the people in their field, not be add-ons to a very organic-centric meeting, but rather have their own meeting. So we applied for this, and as a sort of second-year assistant professor at Caltech, I find myself running this meeting, which was well beyond my pay grade, because I really believed we needed it. We got the meeting. And then I will tell you the thing that was so exciting to me is I sat down and wrote my dream list of bioorganic chemists, who we would now all call chemical biologists. And I said, well, I'm just going to invite all these people, And I left Craig Townsend to do fundraising because I didn't want to do that. And they all agreed. I mean, these were people like Peter Dervin and Jackie Barton and, the greats of bioorganic chemistry. So over the years, the term morphed a bit to chemical biology, but it was that chemical biology term was nowhere near as broad as it is now. Mm-hmm, mm-hmm. It was much more, a lot of things like screening chemical libraries and diversity-oriented synthesis. A lot of the things Stuart Schreiber pioneered felt like they were biochemical biology. But I think then what happened is that term got smoothed out and became all-encompassing. And what belongs in chemical biology is simply defined by the community. And I always challenge the students in my class to give me their best definition of what is chemical biology. And, and they'll come up with things, and they'll come up with bits of the field. And really, it's us using chemical and physical tools to study biology in as much context as we can, as opposed to as much of the model system. So chemical biology, I've taught it for years and years and years, and I've morphed it, and I think there are just some amazing paradigms that one can still teach, even if they were 15, 20 years ago. You can still teach them because they were paradigms. What did people have to know to be able to do this chemical biology? And what could they learn that they couldn't have learned before? So I make a lot about Dennis Doherty's work because I just love it to bits, because that is the paradigm, right? He came from hardcore physical organic chemistry, and all of a sudden he was doing physical organic chemistry on mega receptors in the brain, right? And so he's I'll tell you, he's a personal hero, and every time I hear him talk, he just makes me laugh so much.
There was a fearlessness in his new pivot.
Yeah. And so I think that was very instrumental for me to see that from him, even though he was very modest about the whole thing.
Mm-hmm. Barbara, I want to move to some overall questions about your research. So first, the Imperiali lab, is it a strictly experimental operation? Are there theoretical aspects to your work?
Well, we do things that are not completely experimental. We've dripped in bits of, bioinformatics or computation, more biophysics that's more, quantitative and mathematical than a lot of what we do. So we do cover the spectrum, but over the last decade, I would say, the power of bioinformatics and the abilities to, sort of predict folds and predict functions of proteins has really just been absorbed into our group, because there's so much one can do now through that kind of comparative chemical biology, across millions of sequences, that it's been irresistible to integrate that into what we do. We initially integrated a lot of that work in bioinformatics through my long-standing collaborations with Karen Allen, a true friend of mine who's chairperson at Boston University. And then my group just started absorbing doing this. So those sorts of approaches are really critical now. We have over the years done some computation. We've most recently done some of this because we were doing fairly heavy duty computation of computing the shapes and dynamics of proteins in lipid bilayers. We collaborated with the Straub group and the Allen group at BU on that, but we're all partners, because I think when you do the computational work or when you do the bioinformatics, you need to get a clear-eyed view of the questions. Because if you don't have good questions, you could be just barking up the wrong tree and think that they'll solve your problems. So I'm not a fan of doing those for the sake of. I'm a fan of going in, saying, I have these data. Could you predict it by theory or bioinformatics? And if so, can you then help me develop a model to predict new things that might occur? Sort of always the dialogue between experiment and theory.
Your embrace of bioinformatics and computational biology in more recent years, has that gotten you into machine learning and analyzing the data beyond human minds?
Well, I think once you've got a ton of data, you're going to use machine learning, and it depends on, whether you're using it to figure out what soap people buy or whether you're using it to figure out what amino acids determine specificity. Yes, you could call it machine learning because it's using mountains of data that we can't assimilate, that we need to pour into systems to have it assimilate them for us. But once again, it's really so important to feel like you know how you're looking at the output and what's your input that might vary the output and what parameters do you need to set. So it's so important, even in those sort of big mega projects, that you keep yourself, for me personally, grounded in reality.
Of course, your lab is a fundamental research operation, but I wonder, being in Cambridge, being around all of the biotechnology, being right next to Harvard, have you gotten involved in translational biology to some degree?
Oh, well, I have a small startup company that is located outside of the greater Massachusetts ring. It's out by Worcester, which is based on sensing technology that was developed in the group. Specifically, in the initial phases, it's for protein kinase sensing, protein phosphoprotein phosphatase sensing. So the company, has built on a lot of the IP that I established at MIT. And then we've grown the company. We have about 20 people. We have products. We have sales. We're in that startup hell where you think it looks great one day and the next day, the market took a downturn and it looks a little worse. But I think we're on track. I co-founded the company in 2015, and we've grown up to this size to reasonable revenues, and we hope to be revenue positive. And a lot of that, I think, was really because of us around the biotech hubness of where we are, but in as much as that, I think a lot of the influence on me when I started really realizing the technology I developed could be useful to pharma and biotech were my interactions with bioengineering at MIT, because they are the people who, they see something and they go, oh, someone needs to develop that more. So I gained from that there. Probably at Caltech, it would have been comparable even within chemistry, chemical engineering, because those sorts of people would have been more embedded together at Caltech. So, but at Caltech, I was a little bit more focused, on the earlier goals, where I reached out to colleagues in biology and in chemie. But I think you get the benefit of doing that more when you're a little bit older and you're bringing some different things to the table.
What are the kinds of ailments or diseases for which this research might ultimately be relevant?
So the research, it's really more tools development. So these are tools for people in the biotech, big pharma, academic labs, government labs. They're tools for them to use to quantitatively assess the functions of critical enzymes in cell regulation and signaling. The big top line, if you want to look at the headline, is always oncology, because the enzymes we are able to quantify and study and build drugs to have always gone wrong in cancer, and that's a major focus of inhibitor development for therapeutics. There's about 100 kinase inhibitors actually FDA approved, but the truth is there are well over 500 different human protein kinases, and for many of them we don't have good tools to assess them. And while they may be good targets in disease for therapeutic development, they're needing tools, and that's what we provide. These are tools that can be used in anybody's lab, but can be used in high throughputs by big pharma labs to do drug discovery, to do all the steps of drug development. And one of the big things that we have our sights on at Aslequan, which is the company, is bringing these tools all the way to where they could be diagnostic or personal medicine related, where you could, for example, someone has a solid tumor. Nowadays, they always make what are called PDX mice, where you implant the tumor into the mouse and see how it responds. What you could do with the types of tools that we make is follow progression of treatment of a tumor to see if the enzymes that were out of whack get controlled, see if other enzymes compensate. So really allow you to do much more targeted therapeutics. Now, I said the banner was oncology, but there's just as much interest emerging in neurobiology, inflammation, metabolism, because what kinases are is the traffic lights of the cell and extracellular systems. Go, stop, go.
We'll move now to more technical questions as they relate to your research. the centrality of protein structure and function. What are the big questions today? What's the frontier of knowledge in this area, and how does that compare with when you began your career?
Oh, that just tells you quite how old I am. It was tough. It was tough. Every small victory, purifying a protein, starting to learn about its structure, maybe studying its function, was all done. It felt so painful. It was all very precise, stepwise. It was often reductionist biochemistry, like you made the protein, you studied it on its own, you saw how it behaved, you hoped you could solve a structure. And it was very painstaking. Where we are now with protein structure is the entire universe the structures can be predicted. Maybe they can't be predicted to perfection, but AlphaFold has changed the way people think. We've got mountains of sequence data that can be deployed. And where we really are now, the big problems are, how does everything interact in the presence of everything else, not on its own? Because none of these are solo players. They are actually players that form pathways and networks and higher order assemblies. And that's where we are now, with the ability to do the science at a bigger scale, because really the problems are different from the problems we started working on early on, which was the meticulous stepwise characterization. One of my big interests, and I think is a very important frontier, is life at membrane interfaces. So, maybe 40% of cellular proteins will spend some of their time attached to membranes or embedded in membranes. The rest of the proteome is in the cytosol, which one could call soluble, but there's a lot else going on in there. And what we're trying to understand now is, how do sets of enzymes that work in sequence, how does the membrane facilitate them coming together on a platform and performing these functions with great efficiency sequentially? Because maybe each one of them doesn't work very well in isolation, but how does the metabolome, the machine, work? And for us, the membrane is key because it allows you to bring all these, you've got a lot of proteins that are all floating around in solution. They've got to bounce and interact, whereas the membrane, you are all on the stage. It's when the players come and go, but you're at least all in 2 dimensions. So part of the computational work that we were doing with the Straub and Allen group at BU was really to understand dynamics in the membrane and start to posit questions about, how does the membrane alter these dynamics? What does it do for the reactions? What does it do for the pathway? So that, to me, is biggest problems are looking at biology and networks and pathways, and develop biology, biochemistry, and developing ways to study this where we will bring in anything we can: structure, informatics, biochemistry, measurement, synthetic inhibitors. And we won't restrict ourselves in any way, because all those single topics that I tell you about are things I spent parts of my career on, and I can deploy it into these studies.
The term protein modification, is this to say that it is nature that's doing the modifying, or are you doing bioengineering to modify these proteins?
Well, nature gets everywhere first.
Yes.
Please recall that. So we like to talk about the elaboration of the proteome in the form of post-translational modification. So a gene dictates an RNA, which dictates a messenger RNA, which dictates a linear polypeptide sequence, which will fold. We sort of scratch our heads because there are only 25,000-ish protein encoding genes. I mean, am I as simple as 25,000 proteins? No, I'm not. Because post-translational modification, so I've done all the synthesis, I've made the peptide, I've probably folded it up, is really what explodes the proteome by, 10 to the 2, 10 to the 3 fold. It means we don't just have 20 amino acids in a sequence. We have hundreds of modifications that may be done individually on different sequences to alter the protein structure, the protein dynamics, the protein localization. Like I can stick a greasy tail on my favorite protein called a prenyl group, and it will run to the membrane. It won't be out in solution. I can put a phosphate group on my favorite protein, and it'll do some acrobatics where it'll change its shape and suddenly start interacting with another protein, and then it may go run into the nucleus. So the protein modification is a central theme in biology, only some of the modifications, it took a long time for people to be convinced to study them because they're damn hard to study, frankly. I mean, kinase, putting one phosphate group on, I can picture that in my mind. I decided that was a little bit, too simplistic for me. So I decided to study glycosylation, which is this enormous change in a protein structure when you append the equivalent, and trying to put it into some perspective of a 20% of a protein weight worth of sugars get appended to the protein surface. Now, what does that do to the protein dynamics, or where does it want to live, or how it interacts with other proteins? So we've made a big business of studying protein glycosylation over the years, in addition to the other PTMs, where, I feel like it's just layers of an onion. The more you ask, the more you find out, the more you have to ask to find something out. So the PTMs are important. The enzymes that catalyze PTMs, that do the elaboration, are sometimes hijacked by chemical biologists to put on new groups to proteins in engineering approaches, or we can develop new and different types of chemical biology to do engineering on proteins to change, not necessarily to change the structure, but to put a flag on, like a fluorophore, or to put a localization sequence on that will send the protein somewhere in the cell, or make the protein stick to a membrane. So the modifications are both nature's design and are doing the best we can to co-opt nature's design. The reason I went into this field in the first place, PTMs in general, was I slogged away at a bench as a synthetic organic chemist through my PhD work. And every step I did on a complex molecule, I'd have to have every reactive group protected so I could do chemistry at one place, right? And then I'd do that chemistry. Then I'd carefully peel off a protecting group and do chemistry somewhere else. And that was my PhD thesis. And I said to myself, well, how does nature do all this? Because nature, I don't phosphorylate all over a protein. I don't glycosylate all over a protein. There are short sequences that have the code that say, put the phosphate here if you're this kind of kinase. So I was fascinated by PTMs because I was fascinated how nature did a selective job to execute a selective outcome. And that took me down this sort of strange path, really.
Barbara, you're on record for being amazed at the complexity of glycoconjugates. I wonder if you could explain what glycoconjugates are and why their complexity is so amazing.
Okay, so glycoconjugates just mean biological molecules where sugars, now there's a T.S. Eliot statement where he said, every cat has three names. The name you call him, his secret name, and a third name, right? Well, carbohydrates have three names. They're called sugars, they're called oligosaccharides or saccharides, and they're called glycans. So they're all the same thing. We've just given them different names, first of all. Glycans represent many carbohydrates or sugar molecules joined covalently in complicated structures, branching, linear. Glycoconjugates are when you take those glycan structures and you stick them onto something like a protein or a lipid, which might be in the membrane, or other carbohydrate chunks where you would make what are called glycopolymers. So those are all glycoconjugates, all right? Now, complexity, as chemical biologists, we think of complexity because we think of building blocks. So if you think of DNA, we've got 4 building blocks. We can do a lot with 4 building blocks. We have 4 building blocks for RNA. One of them's a little different from the DNA ones, but still only 4 letters in our vocabulary. For proteins, you have 20 letters, but they can be varied through post-translational modification. So we're looping back to that, and the sequences can be varied. But DNA and RNA and polypeptides are linear polypeptides. There's just, each one is bonded to another one. They're not branched structures. The amazing glycan complexity comes about because there is not this simple, regular, linear polymer, but it's a complex polymer that may be branched. There may be all kinds of different places to form bonds to. So there's way more variety of structure. And in carbohydrates, I will say in humans and mammals, there's 15 or so building blocks, but they're much more complicated than amino acids and peptides. But in bacteria, which is where we focus now, there are hundreds and hundreds of different building blocks. So what's that about? It's like, why did they go off on this? And it's not that every class of bacteria has hundreds and hundreds of building blocks. It's just the diversity amongst all the bacterial species. Okay, so that to me is amazing complexity. It's crazy complexity because there's so much variation possible. Why is it? What does it do? Basically, it's the calling card. It's the language. Single-celled organisms need to talk to each other. They need to make an announcement. I'm so-and-so, and I'm going to interact with that host cell. And all this complexity of carbohydrates is really a barcode that's describing the bacterium to other organisms, other single-celled organisms, maybe viruses, maybe human cells, maybe interactions with extracellular matrices around human cells. So can we deduce this complexity? I mean, I was bold or stupid enough to give a title of a talk I gave recently called Bacterial Glycoconjugates, Making Sense of the Madness. Because what we need to do is start being able to categorize all these different sugars or signatures of sugars or bacterial glycans to try to discern what they're meaning. What is that barcode meaning? So never could there possibly be another good place for AI than that, but we need more ground rules first. So that's the soup we're in right now. It's kind of fun.
Barbara, you alluded to your interest in building chemical tools. More specifically, I want to ask your work on fluorescent and luminescent probes. How do you build these probes and what can they see?
Okay, so fluorescence and luminescence are both really important tools for biology because we don't have, and cells don't have, things that either fluoresce or luminesce very much, apart from green fluorescent protein and that cadre of molecules that were discovered, in the last couple of 3 decades. So we focus on small fluorescent molecules or small luminescent species because they can report the function of processes within cells or in complex media, because you're just observing fluorescence and all the background that might come from all the constituents of the cell is invisible. So that's why you want to use fluorescence. So the technology that AssayQuant is founded on is a small fluorescent probe that allows us to spy on phosphorylation, the activity of kinases, by basically, if a phosphorylation event has occurred, I use an unnatural amino acid nearby and a magnesium ion to coordinate and send out a fluorescent signal. So I can see that event in real time. So what we focus on are small organic chemical fluorophores that give you a lot of bang for their buck by enabling mechanisms of fluorescent signal turn on without being these fairly large, greasy organic molecules that can interrupt a lot of other things. So we try to make probes that are small. They're a little bit Trojan horse. They can you can slip them into peptides and you can use them for assays without creating secondary interactions that will give you off-target measurements. Luminescence is, so that's what we've done with the tools that we use in the company. I have another sort of shoebox of interesting fluorophores that we hope to transition to the company that do what are called environment-sensitive fluorescence, where if one of these small organic probes is within a peptide, for example, and that peptide binds to another protein or another entity in a cell, you would see a light up. So it's a real-time observation of interactions. Luminescence is kind of it's a different sort of tool, and the luminescence is really the big umbrella term, but it's too complicated to go on. But we have worked with both europium and terbium luminescent species. Now, what's different about them to the organic fluorophores? Let me just give you a tiny bit of a primer. So organic fluorophores are small synthetic molecules that you can make in the lab. Lanthanides, such as europium and terbium, are trivalent metal ions of the lanthanide series. And they just have the most crazy photophysical properties because they luminesce with really long lifetimes, and you can do a lot with those photophysics in studying biology. So Karen Allen, who I also worked with on this, we came up with lanthanide binding tags, which are short peptides that can bind to those lanthanides and carry with the peptide that's bound to the lanthanide that can be, this is all, I'm sorry it's so complicated.
No, it has to be, please.
What you can do is you can make your favorite protein with the lanthanide binding tag, and then when you drop in the europium or the terbium, your protein has these properties, these photophysical properties that can be used to study cellular destinations and interactions and dynamics in ways that you can't necessarily do with organic fluorophores. So I just became fascinated in the power of these, but I also realized that there's a lot of fluorophores that are made that are good, bright fluorophores, but they are liabilities in terms of their sizes. So as a synthetic chemist, I made the focused purpose of making small fluorophores that could kind of sneak into macromolecular structures without altering them. They would just be innocent players, except they came along with great photophysical properties like fluorescence and luminescence that could be used to study changes. Is that even remotely intelligible?
Yeah, absolutely, absolutely. So, Barbara, is the ultimate idea behind developing these probes is to understand the role of protein kinases and how they direct or influence cellular activities?
It is more, I think, from the community. Biological tools like, antibodies and different kinds of genetic molecular biology tools where you make knockouts or you make mutations. The biologists have done a splendid job of mapping a lot of this, right? What we need to be in now, so that would be called, the proteomics era, where we're mapping out the structures of proteins, we're starting to understand what they do, where they are in pathways, what the consequences of them going wrong or not being there are, all the mouse knockouts in the world. What we really need to focus on is functional proteomics, and colleagues at Stanford and Scripps, Ben Krabat and Matt Boggio, have all worked on what are called activity-based profiling. So what we need to be doing now is using the sorts of probes that I and plenty of others develop to map out function as it changes, function in time. And I honestly think the most important uses for the types of tools I make are to say, okay, the community has determined that this protein has gone wrong in this disease. If I inhibit this protein and stop its function, do I have a good therapeutic? And what the tools I make are, or have made are, and have deployed into AssayQuant are really tools to help people discover inhibitors, which could be potential lead therapeutics, but also help people find out whether their inhibitors hit 12 other proteins and that would cause mayhem, or are they selective? So by deploying the methods I've developed in profiling, let's check on target, but let's really check off target activity. And so they're much more useful there. So we are in an enviable position. The biologists every day come up with new discoveries or new proteins that have critical functions in certain disease states or developmental states and so on. And we have the opportunity to deploy tools to say, you've shown that that is a critical protein. You've shown that its function has to be tightly regulated. Can we help provide tools that will get you to an inhibitor faster? I mean, this is, sometimes we don't really realize some of these facts, but when you send compounds through drug development and clinical trials, this is years takes. And every step of the way, things drop out. This compound's no good. I'm going to follow this one. When things get into clinical trials, that's when you're really starting first checking toxicity in humans, but then going all the way to testing patients. Only 1% to 2% make it through. And what we believe is if you could make sure your compounds were cleaner early on, maybe that percentage wouldn't be so grim, because all the cost of drug development is at that tail end clinical trials, the billions and billions. The earlier parts are expensive, but they're not they're not as much. So that's really where I think the role my thinking plays.
Barbara, let's go back now, establish some personal history. Let's go back to London. Your degree is in medicinal chemistry. You don't see this as an undergraduate option in the United States. Is that another way of saying you were thinking about being pre-med?
No. Okay, so England is different anyway.
Yes.
All right. So when I was growing up, England, you were tracked pretty quickly after you took what's called the 11 plus. So when you were 11 years old, you had to really do well at the test, or you were toast, frankly. So I had to, and then you go at the age of 11, you go into grammar school or comprehensive school. So grammar schools would be the top tiers, and straight away you choose specialties. So I went into science, chemistry, biology, physics, math, and then I had my cadre of language, religion, English, so on, right? So by the time an English student would get to the end of high school, they would be pretty sophisticated. And I think I had a little bit, sophisticated is the wrong word, I had a bit more breadth of horizons because my father was Italian, my mother was British. We grew up back and forth. I was already bilingual, things like that. So I already had plenty to wrap around the science interests. So I was always interested, even as a kid with my microscope, chemistry kid, in the science, the molecular science. But I didn't like the parts of chemistry that I felt were incredibly dry. I didn't like inorganic chemistry, so I didn't do it. And I didn't really like a lot of physical chemistry, except I need to now for biophysics. And there was this amazing opportunity in the UK in those years, which would have been the mid-70s, where there were 3 or 4 programs. One was at University College London and others were sprinkled around the country, where you could take a degree in medicinal chemistry, not straight chemistry. So if you went chemistry, you had to do organic, inorganic, physical, you were really tracked. Only chemistry. By choosing medicinal chemistry, I kept my horizon open, so I got to do a lot more organic, biochemistry, pharmacology, membrane chemistry, immunology, and enough physical chemistry that I wouldn't make a fool of myself with kinetics and stuff like that. So I purposely chose that track because it represented my ideal, what I really wanted to do. I always wanted to do what I'm doing now. I'm pretty lucky, right? Use chemistry to study biology. And medchem was the thing I could do. If I'd gone into a biology track, it would have been completely away from the molecular. So that was why I chose that option. And then going from the UK to MIT as a graduate student, I sort of made an arbitration at the time that what I needed to be able to do to keep on following my dream, which was to study biochemistry, biology, and so on, was know how to make molecules. So my PhD was in synthetic organic. It was pretty one-tracked, but they let me emerge from my PhD with really strong organic chemistry credentials, so I could then move forward in biochemistry or what became chemical biology.
Barbara, as a student at University College London, this is a generational question and a cultural question. First, how many women were there studying things like biochemistry? Did you ever feel like you were disadvantaged as a woman in terms of what you were capable of doing or where you could end up?
Before I left England, I didn't even think about it. Mm-hmm. I had a strong mother. I actually, strangely, the med chem option at UCL actually had more women than men. Very unusual, because the average chemistry program had way less women. But that wasn't anything that made any difference to me. When I came here, I'm like, well, this is pretty different. We were 1 in 10 women in my graduate class. 60 people, 6 women, 3 dropped out. Those were the numbers. And I kind of felt like I hadn't paid much, naively, I hadn't paid a lot of attention to it. I went to a Catholic girls' school, so my high school was all women, girls. We didn't, I didn't really think that much. I didn't feel boundaries. I didn't feel limits. I started to feel a bit odd about them when I came to the US, thinking, well, this does seem a little peculiar, and we are so few, and why are the men also outspoken? But then I attributed a lot of it to when you do your undergrad, when you grow up in England and you do undergrad in England, you don't speak up in class. Mm-hmm. You don't say a word, And I was faced with classes of students in my first year class at MIT, and chemistry was all these people would fling their hands up and ask questions and answer questions. I'm like, oh no, I'm never going to make it here, because my culture was different. But it wasn't different because I was female. My culture was different, right? And and so it did become troublesome. And I happened to be in the field of chemistry, organic chemistry, where women were really, really, really in the minority. And what felt like happened over the years is that there were great women in organic chemistry, and they all morphed over to chemical biology. So we decided not to put up with all the male and you won't quote me on this, all the male shenanigans and superiority and BS, by moving over to the complexity of chemical biology. Because I personally think we were better at multitasking.
Barbara, when you were thinking about graduate programs, were you specifically motivated to come to the United States?
Yeah, I'd always grown up moving around between Italy and England and stuff. So I didn't feel, I think everyone else in my high school class probably lives within 2 miles of where we were for high school, right? And I didn't feel any of those boundaries. And I sort of decided that I would have a lot more opportunities, because while at high school and undergrad, I didn't feel the gender, bias. I felt an age bias. And to me, I realized that going up through the very hierarchical UK system, where you had to be about 80 before you could be a professor, and you had to be about 70 before you could decide on your own research, that wasn't going to work for me, And I envied the US system. It was much harder, but you could get out there and get independent and choose what you wanted to do. And so that was why I made that transition. I came to MIT. I worked for one of the tough professors because I didn't want anyone to tell me I was shortchanging anything. And then I went on to do a postdoc with someone who was pretty tough as well, because I wanted to be sure I had the credentials for that job that I really wanted, which was to be an independent researcher doing what I wanted to do and not waiting in line. So that was why I did the US transition.
Why
I only
MI,
applied
why
why
MIT? Where else did you look?
nowhere.
And is that
I
because of MIT's reputation in organic chemistry?
I don't know. Well, no, there was one of the lecturers at seems very superficial. There was one of the lecturers at UCL. He said, "Oh, MIT is pretty good for organic chemistry," and I was doing very well in organic chemistry at undergrad. And he said, "You should think about going there because they're good opportunities and so on." And there was one fellow, Franz Sondheimer, who was at University College, a real senior statesman. And he said, "Oh, yes, you should go to MIT, but don't work for George Buchey. He's a tough, mean man. Work for my friend Sat Masamune." I don't know. George Buchey would have been really bad if Sat was a nice man, because Sat Masamune was he was like a samurai. I mean, he really ran the lab like that. So I don't know where I got on this track. Where did I get? Um, so I just, I wasn't of the opinion to go shopping around universities. And I'd been here a summer before. My roommate in a university college was on a year abroad from Mount Holyoke. And so she sort of convinced me that Massachusetts was the place to be. So I thought, singled in on Massachusetts. I thought that Harvard seemed a bit too much like Oxford and Cambridge. And I'd made an executive decision not to go to either of those early on. I wanted to go to the proletariat school, founded by Jeremy Bentham. So I thought MIT was more up my alley. So those are my choices. And then I was really nervous that I wouldn't get in, because at the time I think they didn't take very many foreign students in. And I was filling in the forms thinking, I'm going to put all my chips on this one card, and I'm not going to get in. So I'm filling in the form, and it goes, "What fellowships have you applied for?" I'm like, "What fellowships have I applied for?" I didn't even know I should apply for fellowships. So I went round the notice boards at, this is 100% the truth. I went round the notice boards at UCL, and I saw a sign for the Kennedy Memorial Trust Fellowship, which was a fellowship set up after Kennedy's assassination to bring 10 scholars a year to MIT and Harvard to do to start their graduate. It was only a 1 or 2 year scholarship. And I thought, well, I got to put something on this form. Let me apply for that, so at least I can put it on the form. Right. Sent in the form. Most of the people at UCL didn't really think I was serious about going to MIT. In fact, I had one senior professor come to me in the lecture, because I'd asked him for a letter of recommendation. He goes, "I believe you wanted a recommendation for the MI5. He was joking, of course, because it was MIT. And I'm like, "Oh, okay." Then someone else wrote me a letter, and then the third person didn't bother to write a letter. And then I was invited to interview for the Kennedy Fellowship. And then I got the Kennedy Fellowship. But MIT said they couldn't admit me because there were only two letters on record. And I literally walked into the head of department's office, and you don't do that, and said, "I've got to have that letter because I have a fellowship now and I'm going to MIT." And that was, and the rest was sort of history. But it also got me into Sat Masamune's group because it looked good.
Tell me about Masamune. What was it like working with Masamune? What was the lab focused on at that point?
We were exclusively a synthesis lab. We worked on big molecular targets, and there was a part of the lab that did more inorganic chemistry. He was really, really tough. He'd moved from Alberta. The lab was about a dozen people when I joined. And during the time that I was there, it grew up to like 30 people. So it was a huge lab, and we were in subdivisions, I actually quite quickly got to be sort of one of the leaders in my subdivision, which was a great privilege. But earlier on, I think he was very circumspect about having a woman graduate student from England, because he was very nervous talking to me, and actually didn't really want to talk to me. He would talk to the postdoc standing next to me and tell him to tell me to do something. There was that kind of thing. So I was getting pretty annoyed at the time. And I'm like, I didn't come here to do this. I didn't come here for this. And then there was the famous day, and I've told a lot of colleagues, Masamune has passed on, and I don't think anyone would fault me for telling you these stories. He came up to me in the lab. He actually came up to talk to me, and he said, by the way, do you know how to type? And I'm like, what? Because those were the days you had to type everything on an IBM Selectrics typewriter, and he desperately had this manuscript that needed retyping. And I just looked at him, I said, sort of, but I didn't come here to do that. And he never asked me again. But so he was someone who was really good, incredibly rigorous. I learned so much from him, but some of it was sort of, Wild West in terms of relationships. And the group, of course, was also predominantly male, and sometimes almost predominantly Japanese. Which was another.
Tell me about developing your thesis.
Organic chemistry, especially synthesis, these are not very exciting. They're like dry as a bone. They're so you do an introduction of saying how great it would be to make a compound, and then you have 3 chapters where you're describing the logic and the methods that you developed to make it, And so they're not huge hypothesis-driven inspirational works. They are quite, pro forma, if you will. Like, he gave me 2 weeks to write it. I rented a Selectrics typewriter and went into my tiny apartment in Central Square and typed out the draft, and then read it, and then typed out the revision. So it wasn't any, I have it on my shelf. I tried to make, all the drawings were done by stencil. They weren't done by a chemical, program or anything. It's not, it's only cool to me.
Looking back, how biological was this research? Is this the origins of you starting to think about combining chemistry and biology?
No, to be honest, David, it was a means to an end. It was, so in organic chemistry, to have credentials, like to go forward and write papers and get grants, when you say you're going to integrate organic chemistry into something, you got to have the credentials. So my stint in the Masamune lab was to get those credentials to learn how to do all manner of synthesis, which I did. I finished my thesis work in 3 years, but he had no intention of letting me leave. So I spent 2 years, one as a PhD student and one as a postdoc, a continuing postdoc at MIT, before I did my real postdoc, where I was starting a project between the Masamune group and 3 other groups, the Walsh group, the Petsko group, and the Sinsky group, who were all much more biological. Petsko was a structural biologist, Walsh was a biochemist, and Sinsky was a metabolic biology kind of person. So Masamune had some dreams that he was going to solve certain problems in biochemistry. So he had me kick off a mini subgroup in that area in the Masamune lab, where I basically had 4 supervisors. Walsh, I'd get help on some things, Petsko, Sinsky, and Masamune. So I got a lot more training there in that 2-year interim, and then I went on to do a real postdoc with Bob Abels in hardcore biochemistry.
Masamune's insistence on hanging on to you is really the most important sign of respect beyond the difficulties interpersonally.
Yeah, I think so, yes.
What was the value in staying on for a bit with him?
Well, the value was that it was all new. Everything I was doing was brand new to me. Loads of methods that I'd never done before, like, making protein, purifying protein, running gels. So the value to me was new training and getting it from different groups. So I was really more ready to go on to a postdoc. But the irony of my postdoc at Brandeis was most of my problems at Brandeis and what Professor Abels wanted me to do was synthetic problems. So I was flopped back onto the synthetic path by making certain types of compounds that Abels insisted were going to be terrific, but no one seemed to be able to make them, even the drug companies. So that was my challenge, which I did get through.
What was the initial connection that led you to Brandeis?
Chris Wall, a number of very eminent biochemists worked at Brandeis, either for Bob Abels or Bill Jakes. And Joanne Stubby worked for Abels, Chris Walsh worked for Abels. So it was really it was the happening place for mechanistic biochemistry, which I felt was my next important step, in sort of moving towards my independent career.
Tell me about Abels. What was he like? What was he known for?
He was a great guy. I mean, he was a tough guy, but I think he appreciated my work ethic. When I went for an interview with him, Brandeis is an interesting campus. It's way on the top of a hill, and I think that was part of the sort of interview was, could you get up the hill without being out of breath and then go see Professor Abels? So that was getting there. And then he was much more relaxed, and he didn't care about gender. He didn't care about what people thought or did. You was in it for the science, right? And so I get to the door. His little office was attached right into the labs. And he had this massive poster that he said his kids had given him on the door of his office. And it went along the lines of, yea, though I may walk in the valley of the shadow of death, I shall fear no evil because I'm the meanest son of a bitch in the valley. So I see that on the door and I'm like, okay. And then the first thing he goes, I don't like people who don't work hard. You got to, this is the most important thing. I said, I don't think you need to worry. I worked for Masamune for this many years, so I don't think that. And he, I think he was just sort of betting my personality, and he knew my accomplishments from my record. And so I was there for 2 years, and once again, amazing mentor, tough mentor. People would think that both Abels and Masamune were really, just very hardcore. They were very demanding, but they were both identical. If you worked hard, you had their respect, and that was what you needed to do.
This cemented your identity as a biochemist, your appointment at Brandeis?
I think so. But I still, when I applied for jobs, went for organic and biochemistry with the knowledge that I was going to be doing work at the biointerface.
Tell me about entering the job market. What kinds of jobs were available? Where were you considering?
So this is the bit where I'll go off record.
Okay.
Because my ex-husband was also a graduate from MIT in the more main group in organic chemistry. He was a couple of years my junior. So he had gone to work for Schrock as a postdoc, and we decided to go on the job market together. But in 1986, that was not facile. The minute you tell someone on the phone, there were two of you. I mean, short of hanging up. So we only applied to about 10 places where there seemed to be multiple opportunities, not asking anyone to give us 2 jobs, but just to be aware that there were 2 people looking for jobs. And we just, I did well, he didn't do well. So I got job offers at Yale and Stanford, and then we only got 2 job offers at Carnegie Mellon, which had been his alma mater at undergrad. And it seemed like a great opportunity, and they were willing to put a lot of money in it. And I made the decision. I said, well, we want these 2 jobs, we want jobs together, we're going to go forward. And it was just a disaster because the department basically collapsed. It wasn't a strong department. And then the chairman was caught for embezzlement, and basically the department almost folded, I would say. It was just, so we both decided to start all over again. So I tried to start my career at Carnegie Mellon. I was nibbling in, but when the chairman was caught for embezzlement, we didn't get an incoming class of graduate students, and you can't do very much without graduate students. So then we decided to hit the reset button and look for jobs again, and applied everywhere. I mean, everywhere. And I got one job, and it was Caltech. So we decided, okay, let's try to move to Southern Cal stepwise, because there's a lot of other stuff in Southern Cal. And time passed, a lot didn't work out. So the rest is kind of history. So I got tenure, I think, in 93, and then full professor in 95. And my husband is in academia elsewhere, my ex. So
now joining Caltech to the rescue in 1989.
It was.
Were you following the developments, for example, what the Dervin Lab was doing in those years?
Yes. Oh, it was such a so I was hired into organic at Caltech. I was one of that team. So it was Grubbs, Dervin, Doherty, Myers, and Jack Roberts. Right? I was there with that lot. So proud. I mean, proud wouldn't even begin to explain it. It was amazing, and I mean, Peter Dervin was so gracious. Oh, we're so pleased you're joining us. I mean, it was sort of, they were giving me these accolades that I just couldn't even believe was happening to me after I'd been through sort of quite stormy seas. And yes, so bioorganic, these things were starting to happen there. I taught bioorganic there from the get-go to the upper level graduate, first year grad. And then I also did a lot in teaching organic chemistry there, because, that's what I taught organic chemistry for like 25 years. It's wonderful to teach. Had to give it up when I moved departments, but I feel I did great service to teaching organic at Caltech. Some years I taught with, but Grubbs would go first, and then it was me, and then Dervin. I mean, those guys, we'd go to lunch together every day. It was sort of magical. We'd go to the Athenaeum, and then we'd go back, and sometimes we'd split off and go for coffee. And so that was before Eric Carrera joined. And then Eric Carrera joined, and then there were 6, plus Jack Roberts, the statesman. And Jack was just a remarkable person. So yeah, actually, I doubt you can see it, but I actually got it out. So this, and I can send you a picture of it.
Oh, I'd love it. Yeah.
It was the department when I was pretty new. And in the front row, you had Norman Davidson, then Ahmed Zewail, and Linus Pauling, because he'd come back to visit. So I will send you this, because I pulled it off my shelf. But then back, you see the young Andy Myers and Dennis Doherty and Sonny Chan. And then, people we've lost over the years, like Jay Bailey and Bob Grubbs, the whale. It's Harry Gray was just a great supporter. He'd always make me laugh. He was very supportive as well. So
did the tenure clock reset at Caltech, or was Carnegie Mellon counted?
No, they said, look, we're going to reset it. You may not need it, but it'll take the stress off. So yes, I am one of few people who can say they did their first 3 years of tenure track twice. I loved it so much the first time. And, because I'll tell you a problem, even to this day, if a young professor starts a tenure track that's the classical chemistry tenure track, and they're straight organic synthesis, they can get what they need pretty quickly. If a young person starts chem bio, there's a lag, because you've got to get systems working and going, and complicated systems involving cells and life. In biology, young people often bring their projects with them. So chemical biology, young people were quite handicapped, because they couldn't bring their projects with them, because chemistry departments didn't agree with that. But their projects were much more complex to set up. And so it was a good job I took the extra the extra bit of time. I mean, my personal life was in chaos anyway, so I didn't mind having a little extra time.
Tell me about building up your lab. What was most important to you?
Um, I think I always wanted to have people who I felt were fearless and were in it for the project, not for the next paper or the next this or that. So I tried to recruit broadly people with different expertise. When it came to postdocs, I always tried to bring people in with different expertise so that we would grow in expertise. There was a core we all knew, but people would come in with different abilities and capabilities. So I was able to morph the activities in my group, through that. But to be honest, both at Caltech and now at MIT, these are both places where you can very facilely have a postdoc or a student go to someone else's lab and learn how to do something. I've never felt like I just can't do X and I can't go ahead. I've always felt, well, I'm just going to park someone over there and they're going to learn how to do it. So I've always picked or hoped to pick or somehow recruited people who had that type of, I just want to learn I just want to learn what I need. I recruited by problem, not by what they were going to do, but rather this is the problem.
And by seeing, for example, what was happening in the Dervin lab and the Doherty lab, how did you cement your own reputation in chemical biology? How did that begin for you?
I think we sort of started with flagship work in the glycosylation area, so it was quite distinct. It was more biological in terms of some of the things that you needed to be able to do to make progress in those areas. So I felt that we thought we were very distinct amongst ourselves. So there wasn't really overlapping because Dervin was all in the nucleic acid field. I was in proteins and glycobiology, and then Doherty was in this sort of physical chemical biology, the neuronal work. But before that, he'd been more in the area of molecular recognition and model systems for recognition to understand the counterparts in biology. So I thought we synergized really, really well. And then having really top synthetic chemists like Andy Myers and Eric Carrera as my sort of siblings, basically, gave me, the people to ask if we were having trouble with the synthesis, because we always integrated quite a lot of synthesis into what we did. So I think there was never a feeling that we were overlapping and who's doing something that I was doing. I think we were very distinct.
As a woman faculty member at Caltech, did you experience any difficulties? Was life well in Pasadena in that regard?
Yeah, I never felt, I never felt that there was an issue. We were fewer. I was the only one in organic at the time, but my colleagues were so, so human and normal and irrespective of any of that didn't.
Wonderful. That is wonderful to hear.
Yeah, there was no problem. Where we got clobbered was in the outside world because, everybody needed a certain representation on committees. So we were always asked to do way more than we should have been. It's 10% of the population, but we want 40% women on this committee. So we're just going to ask you anyway,
Barbara, David Baltimore being named president of Caltech in the mid-1990s, the first biologist to lead Caltech. Did that register with you? Did that change chemistry or biology to some degree?
Well, I think it probably changed biology more than it changed chemie at the time. I think it was a big deal, to have a famous biologist come and be the president. I won't say I saw a whole lot of him, but I think it was a stride forward for Caltech to have a president with, such credentials. I think that he didn't understand the chemists as well, as he understood the biologists, sometimes, there would be things that were very peculiar problems to chemistry, and I'm not so sure he had the measure of it, but it was a boon for bio, for sure. But, in the divisional structure, bio was quite separate, really. It was chem, chemie, and bio, and I certainly had friends and colleagues in bio, like Pamela Bjorkman or Steve Mayo, but there was a pretty big chasm between me and them at the time, which maybe it was just X years later when I came here, I didn't feel the chasm, and suddenly I'm in the biology department. I didn't feel that differentiation. I think all the universities have changed, and we blurred boundaries out, all to the benefit.
Did the tenure process change your research at all? Did you feel less bound to do anything in particular?
No, because I was already doing what I wanted to do.
Yeah.
I just wanted to do more of it. Yeah. I don't feel it was like, oh, thank God, I can ditch that crappy work and do something different.
When did you start to think about the big return to MIT?
I don't know. The opportunity came up in 1999. It was, there'd been this enormous, which was not related to my hire, although people decided it was. There was an enormous expose of how women faculty were teaching. I don't know, were being treated in different high institutions of higher learning, around the country. But it was the MIT women who had more or less forced the administration to do a full autopsy on everything, and it was realized that things were not as fair as they should have been. So, and a lot of other institutions, no big surprise, you start peeling off the layers, you find there was inequity in a lot of places. So I remember traveling at the time, and someone at an institution that will have no name goes, did MIT have to hire you because of the woman thing? And I said, no, they hired me because I'm good. So it really wasn't coupled to that. I think they were excited because they wanted to bridge more chemistry and biochemistry, chemical biology. I was an alum who'd done well. So I think there was a lot of excitement about trying to get me back. And I was kind of ready for a move, Some of it was me, my family, there's not a lot of us left, but, my center of mass family-wise is Europe, England and Italy. So LA is an awful long way away. So there was some attraction for me moving back to the East Coast in that respect. And then there was the other part was just sort of sociologically, I wanted to be back in an urban environment. And, Pasadena is a lovely place, but I felt a little bit, it was, two dogs and a cat type of neighborhoods, right? Two kids in a cat or two kids in a dog neighborhood. And I just felt out of place because it was too far for me to move to more urban areas because I didn't want to have some horrendous commute. And I just thought I could have an impact moving, and the cogs fell into place. My family at the time was thrilled because I was so far away, and I saw, it was especially during tenure track years, it was very hard to see that much of them. Yeah. My father would make a pilgrimage to Pasadena every other year, but it was a hell of a journey for him at his age already. So there were just a lot of things aligned that made it a good time for me and appropriate for MIT. But I miss the lunches at the Athenaeum, that's for sure.
These are all external factors you're emphasizing, the community in Pasadena being so far away. Just in terms of the students and the research culture and your faculty colleagues, in that regard, you were happy.
I didn't think there was, there was no big impediment there to getting great science done. It was probably easier because it was smaller and more, a little bit more nurturing. And, the moniker of MIT is IHTFP. And so the students have it all stuck on their laptops. It's I hate this fucking place.
Ah.
It was a little bit more, and it was all the, I thought the moving would shake me up, would shake my science up. Maybe I would ditch some stuff I didn't want to do any more of, and I would go in some new directions. I felt 10 years was good service. I wasn't sort of leaving too quickly. I was, and then I thought, and I was kind of proud to come back to MIT as a full named professor. I thought that was, that meant a lot to me. I sometimes, you feel like, well, it's time to move along. And N years ago, I decided to move along from chemistry to biology, shake things up a bit more as well, my science. So I think it was probably good for my science in all steps.
Moving back to MIT, the plan that this would shake up your science, how did that play out? How did your research change?
I don't really know. I can't kind of put myself quite there. mm, I think it became bigger. I think it became bolder. I think we took on more of the more biological, biochemical. I had some awesome postdocs when I first moved here. I was getting some great postdocs from Europe that I wouldn't have gotten in Southern Cal, I think, because the Europeans have favor the Northeast quite a bit. And the group just sort of grew and was very, very productive. And a lot of the small molecule fluorophore stuff happened here. And I don't think it would have necessarily happened there. I expanded. So, I was mostly doing the glyco work at Caltech with a little bit of biological probes. And we sort of expanded the probes in full measure. And that's what became all the sensor work, fluorescent, luminescent work we'd done. So I think it just expanded the footprint of part of the group. What did I leave behind? I can't really remember. But they all moved with me, which was a really impressive.
Now you're experiencing MIT through a very different perspective now as a member of the faculty, not as a graduate student. Still, I wonder if you could reflect on how the culture at MIT changed or did not over those years.
It was just felt like a different planet being a faculty. Yeah, I know. I mean, I always joke that the main thing that changed is I could afford to buy lunch, but it was a little better than that. It was really a different place. I mean, some of the old faces were here, but it was a very different place. The building was all renovated, and there was a lot less of the hardcore organic. There was more biological, and then there were these opportunities to form, allegiances with bioengineering and biology. So, I would have never strayed beyond my lab on the third floor of Building 18 when I was a student. Now I'm sort of, around.
And the transition to becoming more biologically oriented, was your work in protein glycosylation sort of the pathway to that?
It was a necessity to do more biological work because the problems could only be addressed through more biochemical or biological approaches. And the biological significance of glycosylation, I will tell you, it was really dark ages stuff when I started working on it. A lot, there were, it's like, oh, don't work on that, it's too complicated. People have realized by leaps and bounds the significance of glycans and glycobiology in biology. It's not well represented because you had to be half mad to take it on. But it really, over the time I've been in this field, it is just, accelerating the roles of sugars in biology.
Tell me about how you got involved in ribonucleotide reductase work.
That's not me. I don't think I had any papers there. That was more Joanne Stubby.
But this was an influence for you.
Why did I ever say something like that? Joanne Stubby is a senior biochemist who was in the department when I got here, and so she was, very, very strong biochemical figurehead in biochemistry in the department, and taught us all to take no prisoners, shall we say. But so one of her central themes was ribonucleotide reductase, because it plays such a pivotal role in DNA synthesis. It's taking that oxygen off the ribose. But I've never worked in those areas.
The transition to biology as your primary appointment, was this a gradual process? Was it one particular paper?
It was a mixture of science, personal decision, again, different environment, again, to do the change in department.
And what was the research? What really compelled you to sort of make that shift?
So, it wasn't so much the specifics of the research. It was being more surrounded by people who were talking about different biological phenomena or things they'd discovered or they were working on. It was more, what does the world at large need from chemical biologists? It was much more the community, to a certain degree, dictates or bringing to your attention new things that could be going on, new knowledge about a lot of the techniques that are so prominent now in biology. Whereas in felt, to some degree, we were starting to get a little isolated. And so didn't want to move, was happy on the campus, and so I made the transition. And it did happen quite quickly, really.
When do you start thinking about DNA damage?
That was some work that we did with the in relationship to all the work that we were doing with kinase assays, but we were also working on strategies for developing cage proteins that you could turn on and off a function. So we did some really cool work with the Yaffe group, who is in biology, actually is in the Koch Institute, where we were developing chemical biology tools to study the DNA damage response. So that was where that fit into. It was a demonstration of the tools we were making. Another influence I had on that is I joined what's called a UO1 grant in cell migration. UO1's a big multi-PI grants that Doug Loffenberger and Linda Griffith in bioengineering spearheaded, and they married me into that big initiative where I could do probe development, but be amongst the biologists who studied cell migration. So, it's still an issue with even NIH funding it's still very much hypothesis-driven work. And if you're making tools, you need to be applying them to do hypothesis-driven work. But making tools takes time in the first place. So you need money to do that before you can do hypothesis-driven work. So on the UO1, I had the opportunity to develop some of the tools and apply them into the cell migration work. So in several cases, with the probes work, with the sensors, I've taken tools I've made and collaborated where through meetings or seminars, I found ways to deploy what I make into other people's research where the tools that we make could ask more incisive questions.
Barbara, when do polyprenols enter the picture for you?
Okay, polyprenols are strange molecules. They are long, big, greasy molecules. So you know about lipid bilayers, okay? Long chains, they come face to face, they make a boundary, great. Polyprenols are much larger than the tails on the lipid bilayers, and they're used, the really long linear polyprenols are used exclusively in glycan assembly, where that long tail parks in the membrane, and all the enzymes that sequentially act to build up a glycan do it on a polyprenol phosphate. Why is that? So very early in my career, where I started studying glycosylation, I thought, well, first of all, I'm going to figure out why polyprenols, and I'm still here doing that. So what we believe, and I think that will be a nice capstone as I move towards the later years in my career, is that we believe we're starting to get testable hypotheses about the roles of the polyprenol in these multi-step pathways that occur at membranes. Because we think that the polyprenols may help create a microenvironment where enzymes that all belong to a pathway congregate in order to function sequentially to deliver a glycan product. And the polyprenol is there really as not really a chaperone, that's the wrong kind of word, but it impacts the structure of the membrane in ways that may promote diffusion and interactions of enzymes involved. So the polyprenols had to feature early on. I needed polyprenols. They were devilishly expensive. I discovered from papers that there was a high concentration of polyprenols in ginkgo leaves. I went out with my first graduate student on the streets of Pittsburgh to collect ginkgo leaves to extract them to make polyprenols. There needed to be a chemical reaction to convert it to the dolichols, which are the ones that are used in mammalian biology. We developed some chemistry for that, and then we moved further on. Then we studied a bacterial polyprenols, which are slightly different from the mammalian ones. It's a funny story, so you'll get the punchline.
Please.
Well, they don't come out of ginkgo leaves. They come out of the dried red leaves of staghorn sumac. So I returned to my roots, went collecting leaves, extracted those linear polyprenols to do my research, because I don't believe anything should get in the way. And so there's a thing. So, but they are pivotal for the biology and biochemistry. But, you sort of feel, gosh, I can't believe we still haven't solved this problem. I still can't believe we don't understand. Nature doesn't do anything without a darn good reason. And I think the reason is very much, though it looks like a structural entity, and it is in a membrane, I think it's functional with respect to the impact that it has on the biophysical properties of membranes to promote reactions. So it's not a bystander. It's not just an anchor. It's functional.
Barbara, arriving in Cambridge in 1999, it's 25 years later. When does biotech transfer from being just a few buildings here and there to this enormous endeavor, making Cambridge a hub? And did that affect your research at all?
While I was gone, it happened. Cambridge was not the same place when I get back with respect to that. It just grew and grew and grew. I think the thing about being in such a hub is that there's just a lot more activity around. There's lots of people coming and going. Your students can find good jobs in the area. There's just a lot of interest. Seminars can be, you'll see industry people come to seminars. So there's just a general heightened interest in the biohub. It's a little bit over the river too with the Longwood campus, but there's so much in Cambridge. I mean, I can look out the window and see, different companies, Novartis, Biogen, and so on. I think it's great for our area. It makes parking a bit difficult, but I'm okay with that.
Barbara, we'll move our conversation closer to the present. When COVID hit, did you see an opportunity to become involved in coronavirus research?
It is so silly. There was a lot of the early papers, I got really angry. There was some early hypotheses about this, that, and the other with COVID. So I didn't get involved in COVID. We kept on chugging. But what I did try to do a little bit is say, guys, we got to look at the sugars because the spike protein is covered in sugars. And there were people making statements about mutations in the virus, the spike protein, and how each new variant that came along had this mutation and that mutation. Once you make mutations in the protein, the sugars jump around. And I felt that I actually tried to call certain people who were putting things on bioarchive and said, don't you think you should be talking about the glycosylation of the spike protein as well, because it's pivotal? And, people were uninterested. I would say despite the fact that glycans are so important in virology, they kind of get short shrift because viruses use our machinery to make the glycans. So we think that the viral glycans are just the same as our glycans, but there's actually subtle differences. So I felt in the COVID time that there was insufficient, maybe, I think it grew after a while when people were becoming desperate to sort of characterize the spike protein, which was what they were making the mRNA to make the vaccines from. And so you really needs to have the grip on the glycosylation patterns because they will influence the immunogenicity, which will influence the response.
During the mandates to do remote work, had your lab become sufficiently involved in computation and bioinformatics where it was a relatively easy transition to be away from the lab?
It was fine for some people,
Yeah.
people who had data sets and were structural. It was tough for others. So we sort of went on a kind of mission for everyone to do another round of learning. So everyone was presenting different topics that they had to prepare. And then we did do quite a lot of writing in that time and just begged and pleaded. We finally got some access by June. I'm not running a big group, so it was not that I had 25 people to deploy. I had about 10 people to deploy. And so, we survived, but it hurt, And our biggest job was not worrying so much about the science, just worrying about keeping our people safe and sane.
Yeah.
So creating enough opportunities for things to do, to interact with other people, even if it was on Zoom and so on. It was murder.
Barbara, we'll bring the story right up to today. What is the Imperiali lab currently focused on? What's most interesting to you in the field?
We are focusing on the aspects of the bacterial glycan pathways. So, as I said, the making sense of the madness, trying to develop some paradigms for if you've got a genome, can you predict the glycome? Skip the guys in the learn about the proteins in the middle. And then the other thing that we're very excited about is we've had some developments lately in making glycan binding proteins. So if you want to study a protein in a cell or in an animal, you make an antibody to it, you can use it, you can probe, you can do all kinds of things. There's also therapeutic antibodies. If you want to study DNA and RNA, you capitalize on duplex formation and being able to sequence and synthesize. If you want to recognize glycans, you're clean out of luck because the predominant proteins people use to recognize glycans are things isolated from plants, fungi, and some bacteria that are called lectins. And they're weak binders. They're not very specific. They won't make any sense of the madness. We don't have near enough of those. You can go to huge lengths and make antibodies to sugars, but they're not very antigenic. It's expensive. You have to use animals. So we've been developing yeast surface directed evolution of small scaffold proteins and demonstrating that we can make selective glycan binding proteins out of those that can ultimately, we hope, be integrated into what we call glycan readers. So if you have a small glycan binding protein that recognizes a couple of sugars and then another one that recognizes a couple of other sugars, can we ultimately make readers to read a bigger chunk? So that is something I want to push hard enough to get to commercial viability because it's so badly needed.
So this is really a connecting point between the lab and the startup.
Yeah, it is. Yeah.
Well, Barbara, for the last part of our talk, now that we've worked up to the present, I want to ask a few retrospective questions about your career, and then we can end looking to the future. So first, if you'll indulge me, just one question on some of the most significant awards that you've been recognized for. Both because of where you come from and where you are now, I wonder if you can compare the honor of being a fellow in the Royal Society of Chemistry and being a member of the National Academy of Sciences.
Oh, the NAS is such a big deal. You see, I'm not Unfortunately, I'm not I'm only in the chemistry part of the Royal Academy, not an academy member. It's different. There's a hierarchical thing. So the NAS is like the big deal, and I was very very proud, and I think even my relatives were impressed when I was elected to the National Academy because so that opportunity. So that to me, that was, I would say that was pretty significant. Uh, Awards, you have to be fortunate enough to have people who want to put you up for them and want to be your advocates and get you in. And I genuinely believe that, even though I was here, I think Harry Gray was really my advocate. I suspect it because he was so happy about it all. So
Barbara, other awards honor your commitment, your love of teaching undergraduates. This is not an automatic for professors at research intensive universities. Why is teaching so important to you? Why do you get such energy from it?
I just love explaining stuff. I mean, I love using quirky ways to explain things. I love getting on the level of the student. Nothing gives me more satisfaction than if I've taught a good class. I love getting them engaged. I'm excited about what I'm telling them. I'm giving them my insight in chemical biology, or I was doing general biology, I was doing organic chemistry. I just enjoy it. I overdo it. I spend infinite amounts of time to find the perfect picture or the perfect description, but it's just something I just get a lot of joy out of. And I think it makes a difference, especially in some courses where, OChem is the pariah, nobody wants to take OChem. But, I taught it at Caltech, I taught it at MIT, and I think I had a lot of students who loved the course once I got them over the fear of it. Uh, so that is my most proud achievement is the Feynman Award at Caltech. So forget all the other awards.
That's the one.
That's the one. Yeah, that was
Barbara,
really.
all the graduate students you've had, all the postdocs that you've worked with, what has changed and what has remained constant in terms of their interests, their motivations, what they see as their opportunities in their careers?
I think, I think there was more optimism earlier on. I think the sky was the limit. I think there were a lot more opportunities. I think in this last decade, it's felt like the students and postdocs just were worried all the time. They were worried that we were struggling to get grants, or they were worried about jobs, or they were worried about getting to the end of a paper to get a chapter for a thing. They're a much more nervous crowd right now, um, and it's fearful it's worrisome for our community because I felt that they were much more sort of, I'm just in this because I love it. And now I just feel there's so many pressures and stresses on students that, are we scaring them off? What is it? I don't know. I don't know how there'll be a rebound because the amazing thing about science at the university level is you don't know what you're going to find out often, and it's discovery and it's serendipity. And if you're so worried about, who's going to cover my dental plan, it's a huge restriction on creativity. Don't get me wrong, you still got tons and tons of good students, but as a whole, there's more nervousness and worry. And, because I think it's a tough, it's been in the world now, right? Wars, climate, and everything. There's a lot more on their plates. And how could they not have changed?
As you interact with your colleagues who are junior faculty members, what are the pressures that are facing an assistant professor today that might be different than what you faced?
I think when I was assistant professor, we didn't worry. There were so many things we didn't worry about. We didn't Now the students are all unionizing. So, for example, our lot here unionized, and that's made a lot of difference to our junior people because you didn't used to be, well, nobody let me keep a time card or count vacation day. We didn't do any of that. Maybe I had a health plan, probably, of course, but we didn't have all the worry. We didn't have all the things that we had to check the boxes of as PIs. Students came, they were thrilled to be there, they were getting a living wage, they were excited about science. Now our junior people have a lot of things they have to worry about. So it's much less unrestrained love of going forward. I mean, if you got a good enough, if they're good enough, they're going to do fine. I just feel that it's awful to say, and maybe there's a better way to say it, but it seems a lot less joyful right now. And I don't want to end on a downer here, but the times have changed.
Barbara, your work has been lauded in a very interesting way for your capacity to look at proteins in their living context. I wonder, what does that mean for you? What does it mean to see a protein in its living context? What are the tools to do that? And what can you see that is not possible by looking at a protein in its dead context, as it were?
Well, I mean, I think all biological systems are all very complex communities of macromolecules interacting. So what the most important thing to do is to try to look at them in context of what else is around when this protein functions, because in almost no cases are things singular. So we try to develop tools where you can look at functions more in context than what we would call in vitro. So you can go from complexity. You can go to a test tube of clean protein, right? Nobody else is there. Buffer. Then you can start adding stuff to the mix. But then you can take a chunk closer to a cell by using the lysate of a cell or the lysate of a tissue. Then you can go in live cells where metabolism and everything is still happening. There's a lot of people who are far better at that than me. And then you can go to organisms. So I think what's important, what we all recognize, is that you can't do reductionist biochemistry anymore and learn anything about living systems, because you have to throw in the complexity, and the complexity makes everything more challenging. And the complexity with looking at systems that work at a membrane, I mean, you can't solubilize your protein out of a membrane and expect to see anything happen that looks anything like the real thing. Because it's like you took dish soap and you pulled your protein out of the membrane. It's not the same. So having to attend as much as possible to the native environment of proteins and reactions and interactions is the thing that I think is most important.
Barbara, I'll use a title that you came up with for an article as a way to capture what your research represents: interrogating biology with a chemical lexicon. I wonder if you were getting at something deeper than just using chemistry words to understand biological problems. I wonder if you can reflect on that.
I think I was trying to bring in what I always try to do is say, the biology field is very, very fundamentally important to life, right? But I think that what I wanted to bring forward was that chemistry has a real seat at the table to do that. There are things that we can interrogate with chemistry that we might not be able to do with genetics or molecular biology or immunology or other classical methods. But more and more biologists are realizing that, they're using some of these tools like Boncat, things that Dave Tyrrell introduced. They're using it, they're realizing. The trouble is we need to educate them enough about chemistry to realize that all these tools and reagents, if they don't work the first time, you've got to tell the person who made them so they make it better so it works the second time. So this disconnect where people will buy a reagent from Sigma or somewhere and it doesn't quite work and they say, oh, this chemistry is all rubbish, And you really got to form partnerships, because coming up with the chemical lexicon to interrogate biology means an engineering process where you go back and refine and go back and refine until it's good enough to be in anybody's hands to ask a particular question. So I think what I was really trying to speak to there was the need for chemistry in the life sciences. And sometimes I think the chemists are going to get chucked off the bus here, and it's really important that they stay on it. Wasn't the feeling I got at Caltech, but just in general, there's departments going, oh, who needs chemistry? You need chemistry to understand life.
Well, finally, Barbara, looking to the future, if we can zoom out in the grand sweep of history, if you're really part of the founding generation that has interrogated biology using a chemical lexicon, where does the field go from here? What's the frontier as you look at, undergraduates who are captivated by this, graduate students and postdocs who want to make their own contributions beyond the timescale of your own career? What are the big problems that they're going to need to look at?
I think the sky's the limit, but their biggest problem, not big problems to look at, their biggest problem is integrating everything that's available to them and maintaining enough of a level of sophistication to know what's coming out that's going to make a difference to what you're studying. We desperately try to keep up. So I think, so many challenges can be tackled now that you couldn't have dreamed of 10, 20 years ago. It's a wealth of choices, and I think the biggest challenge is finding those incisive issues that if I knew this, then I could do that, finding those, but then being prepared to deploy any method you can to get to the bottom of it. Is it chemistry? Is it physics? Is it mathematics? Is it biology? Because you can only tackle the brain, right? How are we going to figure that out? Me remembering, to brush my teeth in the morning, how many processes are going on, right? So those are gigantic problems. And it's the old, you've got to unravel it all to understand it, in certain cases to enable the recognition of approaches to disease or ways to prevent disease. So I think it's really, it's an embarrassment of riches, and people will just have to sort of be prepared to be very aware of the opportunities. And at the same time, they're going to have to be careful, because I think even in my day, it was like, you really got to be careful that people don't call you a jack of all trades and a master of none. If you're, one day doing this method, one day doing the other, you've got to prove to them, no, I've got my credentials, I'm doing this. Therefore the synthesis thing.
And just as an addendum to that, as you survey your own career, your own research accomplishments, you've articulated it's a problem. It's an intellectual, organizational problem to look at all of these things. How do you view your work as a bridge to these future discoveries?
Yeah. Well, I don't know. There's plenty of people who are far more famous than me that make far more impact. But I think it's just showing what can be done with a complex problem where you start and you really don't know where you're going, but that you're prepared to work your way through it. So the bridge is really, if I were to write an autobiography, would I be able to demonstrate that everything was leading somewhere all along and that I had it in my mind? Possibly, but in some cases you're just lucky.
And there's some grit in there too.
That's too much grit.
Barbara, this has been a wonderful, wide-ranging, so interesting conversation. I want to thank you so much for spending the time.
Yeah. Well, thank you for your patience and interest.